Hello everyone, you're tuned into today's PIR live event, and I'm your host for today, Scott Jones. Our guest today is Dr. Diane Lagace, who is an assistant professor in the Department of Cellular and Molecular Medicine in the Neuroscience Program, Faculty of Medicine at the University of Ottawa. Dr. Lagace is going to help us understand how adult brains make new neurons. So welcome to the PIR live event, Dr. Lagace. Before we get started, I'd like to also welcome today's viewers who are watching in on the live stream. It's great to have you with us today. Remember that if any time you have a question, you can tweet your questions to us using the hashtag AskPIR through any time during the event. If there's room in your tweet, please include your name, school, or city so we can give you a shout out. I look forward to getting to those questions, but first I'll turn it over to Dr. Lagace, who's going to tell us a bit about her background and then talk about her research in adult neurogenesis. So thank you so much for joining us today, Dr. Lagace.
Thanks so much for having me, and it's a pleasure to be here, and I really appreciate that all of you have taken the time to hear a little bit about what we're doing here at the University of Ottawa, and specifically in my lab that's studying the birth of newborn cells in the adult brain. So I'm a researcher here, and I have a lab at the University of Ottawa, and we're trying to understand what are these new cells doing in the brain. So how did I get here might be one of the first questions you have. And what I did, first of all, is I actually was a student, a high school student, maybe like many of you, first of all, in Toronto. Then I went to the University of McMaster in Hamilton, where I did my undergraduate degree in psychology. I then went all the way actually to the East Coast, to Dalhousie University, where I did graduate work in pharmacology and also worked in psychiatry. And then I moved from there down to the University of Texas Southwestern, where I did a postdoctoral fellowship. So that's sort of like a residency for medical students. And basically what you do is you get to work in a big lab and try to really understand how does the brain work. So I've been studying neuroscience for many years now, and it's a pleasure today to share with you some of my work and what my lab is working on, really trying to understand how new cells are born in the adult brain. And I think the best way to do this is to show you some pictures and get you a little bit engaged. So I'm going to now open up my PowerPoint slides for you. And this way you'll be able to follow along and look at some pictures while I look at them also. Okay, can everybody see? Everything's good?
Yeah, it looks good on my end. Go ahead.
Okay, great. So this is a picture actually of newborn cells in the brain. So all those pink things you see all around here are all the newborn cells. Okay, and you're going to, by the end of my talk, be able to understand where these cells are, and you're going to be able to, if you see a picture like this again, you'll know where you are in the brain. So what do we know about these new cells? Okay, I'm having a little trouble advancing, which isn't good. Oh, okay, just 1 second. So I go browse by individual, right, to get it to work correctly, and then OK.
Yeah.
And then play slideshow. Okay, now I'm going to share it.
Okay, I've got the first slide again.
Okay, here we go with the next slide, right? All right, now we're in it. So for a long time in the brain, we thought that there was no new neurons could be born. So this is a picture actually from a scientist, Raymond Cajal, a very famous neuroscientist in the early 19th century. And what he said is basically, once your brain formed, you couldn't make any new neurons. So what he says here, you'll see in yellow, everything must die. Nothing may be regenerated. And it's for the science of the future to change if possible. So this is what our lab is working on. We're trying to understand how can we make new neurons in the brain. So for a long time, and even in some of your textbooks right now, what you might think is all the cells you have in your brain are born when you're first developed. So even starting from your embryo, this is what you looked like. You initially were just 2 cells, then you became 8 cells, and then you became a blastocyst. So you formed from just these few cells, and all those neurons were derived from those cells. But what we now know, this is a picture of the human brain. So what your brain would look like, if we just opened up and looked inside of you, we know that there's new neurons being born within that brain. at all time points during your life, which is really incredible if you think about it. Imagine that at any time point you have right now probably a new cell being formed. So this was first discovered in science in actually the 50s and the 60s, 1950s and 60s, and it was first shown in mice and rats, where studies showed that in these species you could see brand new neurons being born. It was then discovered in primates in the late 1990s, and then deemed the discovery in humans. So the first paper was 1998, and it came out in this journal called Nature Medicine. So for those of you who don't know what I'm talking about in terms of journals, what journals are is they're like kind of like medical newspapers, and now we see them online. And they give us what's the most important scientific news to share with the whole rest of the world. And the title, when it was first shown in humans, was Take Comfort in Human Neurogenesis, okay? And in this paper, they showed for the first time that there was a birth of new neurons within the adult brain in humans. So why is it so exciting? Why would they, if you have to think for a second, why do you think that it would be so exciting that there's new cells born in the adult brain that can form new neurons? So the neurons are the cells, right, that talk within our brain. So why would that choose so much excitement? And why would people say take comfort? Why would that be the title? So it's a little hard sometimes to be able to get feedback during one of these talks, but I want you to just think for a second, if you can think of any reason why people would be so excited about this, because let me tell you, when this happened, it was in the New York Times newspaper, it was all over the news, everybody was really, really excited. So the reason is because what we know is that because there's new neurons being formed, and that process is called adult neurogenesis. So you'll see there in red the words adult neurogenesis. So that's the birth of new neurons. Those new neurons that are born, they're very important actually in terms of understanding how normal brain works. So in you and I and everyone else, we know that these new neurons are probably really important in our ability to understand how we learn new things and also how we remember things. So we actually, even though neuroscience knows a lot about the brain, we actually still don't know how we learn things and how we remember things. And the other thing that's really important is that there's a lot of diseases out there. So like Alzheimer's, you might know people that have Alzheimer's disease or Parkinson's disease, or someone that has a spinal cord injury, or someone that has a stroke, where you actually don't have very many neurons anymore, or that you've lost neurons. So can you imagine if we now know that your brain can create new neurons, maybe those new neurons can be used to try to fix those diseases. So those neurons could be used then to help people with Parkinson's disease, or with Alzheimer's, or with stroke. So why is it that it's important in learning and memory? So those new neurons, actually, one of the remarkable things about them is that they're only formed in a certain area of our brain. So in the top left, you can see a picture of the brain. So that's the human brain, but it's cut right down the center for you, okay? And then it's opened up so that you can see the structures on the inside. And where that line is, so you'll see a line through it there that has a 3 above it in the top left of your screen, or right maybe. What they've done is they've cut it and turned it so it's showing you this picture of the brain down here. This is called the coronal section of the brain. And this little blue region right here, right deep within our brain, right at the bottom, is called the hippocampus. Okay, so that's Latin actually for seahorse. And that hippocampus, we know, is really important in learning and memory, in learning new things and remembering things. So in all of us, that hippocampus is very important. And you know what's remarkable? If you look at that whole brain section, so that big brain section you see, and there's some other parts of the brain labeled, you might know some of these, that outside part that's all got all those curves and crevices in it, that's our cortex, right? But right only in that hippocampus is where those new neurons are born. So isn't it remarkable that actually in the adult brain or in teenage brain and in many different ages, the only spot in our brain, or one of the only spots, is the hippocampus. And that's why we think that these cells are really important in learning and memory. So how do we know that those cells are there? How can we study those cells? And what you might be aware is in biomedical research at the University of Ottawa and many places around the world, we try to understand these new cells by looking at different types of model systems. So we can look at cells in a dish. That's a picture of the person with, you see the red liquid within a dish. So we do this in my lab, and we put those cells in a dish and we can grow them and see what happens to them. There's other labs that study worms, and worms can learn new things. Flies at the bottom. And then also we have a lot of animal research going on, as well as studies within humans. Now, in the study of neurogenesis in a human, we can't actually see in a live patient those cells. They're just too far deep within our brain. So what we have to do is look post-mortem, so after someone has died, to be able to understand what happened maybe to their newborn cells. So since we can't look in the live living brain, right now what we do is use animal research to be able to understand what those cells are doing and how can we better utilize those cells to treat disease. And I just wanted to put this slide up there because I know some of you might right away think, oh, I don't want to ever use animals in research. And I can understand some of your concerns with that. And when I was younger, I also had concerns. But now that I work in the field, I really understand that there's a lot of myths out there about animal research. And what we're doing is really more to benefit human health and also to understand diseases. So the question that one of the questions we're asking, and that in the blue is a picture of the mouse brain, is we're trying to understand how can we change the number of new neurons that are formed in that hippocampus. So all those black little circles that you can see are the newborn cells on the left. And you can see there's a lot more of those dark circles, right? Those are the newborn neurons, okay? But on the right side, you'll see that there's only one or two of them. There's not as many. So what things actually make more or less newborn neurons? So what do you think happens as you age, for example, or as you get older? Do you think you have more or do you think you have less? What actually happens is you have less. So as someone ages, you produce less new neurons. What about if you exercise? So lots of us enjoy exercising. Do you think you have more or do you think you have less? You have actually more. So if you exercise, you can create a lot more newborn neurons. So what do you think happens after a stroke? So this is a picture of a human brain post-mortem after a stroke. All those dark areas are where the cells have died. And what happens in a stroke is you actually have a loss of blood flow to that area of the brain. So those neurons all died off. So what do you think happens? It's quite remarkably, what you have is actually more. So this was first shown in guinea pigs, actually, in 1998 after a stroke. So there's the top is a control with no stroke, and the bottom is with a stroke. So why would the brain make more cells after a stroke? So those new cells actually go right into where the damage of the stroke is. They divide and they migrate right there. So you might say, well, if the cells go there, then why doesn't someone get better after a stroke? What is it that's stopping someone from getting better? What we know, sadly, is that actually about almost all of those cells, so 80% of them die during their development in the stroke brain. So they divide and they go to where the stroke is, but they can't make a new neuron. They can't connect in and actually help to save or to restore function in that area of the brain. So it's sort of like this diagram here where you have lots of cells in a bottle, right? And they're all going to that empty bottle that is where the stroke region is. But in the end, only that little one blue-green cell actually ends up surviving and becomes a new neuron. So we have lots of potential there, right? There's lots of cells initially that could help, but actually very few of them survive. So this is the research question that we're working on in my lab. Our lab works on if we enhance or make more of them survive, can we improve stroke recovery? And we can model this in animals. So these are some transgenic mice models, and this is post-mortem, but you can see hopefully you can see this is the site of the stroke up in the top of the brain where I have the word core is where the stroke is. And the cells are all those green dots actually, and look at them all, they're all going towards where the stroke is. And we actually have some special mice that we've manipulated to be able to see whether we can make more of those cells survive. So if we can make more of the cells survive, what do you think will happen to the animal's function after a stroke? So to look at function or what happens to the animals, we actually have, just like in a human hospital where you go to rehab and you can measure how much are you getting better after a stroke, we have an animal behavioral core facility where we ask and look at how does the animal behave? What does it do after the stroke, and does it get better if we give more new neurons? So this is just one example of that simple task. How can you test that in a mouse? So you can see this mouse here has a little piece of tape on each of its paws. And what it's going to do is actually try to take that tape off its paw. It doesn't really like the tape on its paw. It doesn't hurt it, but it doesn't prefer not to have tape on its paw. So if you stroke one side of the brain, it's not going to be able to take that tape off its other side of the brain. So off the opposite limb is not going to be able to take the tape off. So it's a little bit busy in this graph, but what I'm showing is the time to take off the tape, so the latency, at different days after the surgery or the stroke. And what you can see is that after a stroke, the animals take on the paw that's opposite to the side of the stroke, almost 30 seconds to take off the tape, where most animals are, doing it very, very quickly. Within 1 or 2 seconds, they're able to take off that tape. So we measure how does the animal behave afterwards. So all of this work that I've talked about to you today is done within my research lab, and these are just some pictures of my research team. And really in research, when you have a team, it's almost like your family in some ways. So I have different students at different levels. So I also have a technician. This is Karen. It's in the middle of, I don't think you can see my cursor, which is too bad, but in the middle of the picture is Karen, who's one of the technicians of the lab. So she has a master's degree in science and now works in the lab full time. There's also a variety of students that work in the lab. Some of those students are PhD students. Some of those students are medical students. Some of those students are high school volunteers. Usually each summer I have one high school volunteer in my lab. And then you can also see my children in that picture. They're almost like part of my lab too, although they're not allowed in the lab because they're a little bit too young. But they're also part of our research family. The work in my lab, why we're able to do this is we get a lot of funding from different agencies in the government. So some of those I've shown, those at the bottom. And also we're very lucky in Ottawa. I just want to point out that we have, and actually it's now a national Center for Stroke Recovery. And this center allows us then to try to understand how can we make recovery better for patients that have a stroke. So what I've hoped you've learned from this, and what I'm hoping you're going to take away, is that you can make new neurons in your brain. So right now, new neurons are forming in your brain. And probably more of them are in your brain than my brain, because I assume most of you are younger than me. Those new neurons also will help you learn and remember new things. And we also know that after a stroke, even though you might have thought, I'm sure some of you thought that there was less new cells, there's actually more new cells dividing, but most of those cells actually can't survive. And so we're trying to understand, our research is trying to understand how can we make them survive, and would that increase or improve recovery from stroke. So really this leaves us with a lot of hope for the future. I believe in the study of neuroscience, we're going to make a difference, and we're going to be able to help a lot of patients and people that we know. You might know people who have diseases or different disorders that are associated with less new neurons. So we're saying, can we make new neurons throughout the lifespan? So can we make the brain more plastic? So this is a picture of clearly an embryo, a young child. There's lots and lots of new neurons being formed there. But as you get older, there's less neurons. Remember that? Remember I said there's less neurons being developed. So we want to know, how can we make more of them survive? So that gives you a little bit of an oversight into what we're working on. And I appreciate very much that you took the time to listen in. And I know I went kind of quick, but I like to show a lot of pictures to give you a chance to be able to see some of the images from the lab and things we work on. And I welcome any questions that you might have.
Well, thank you very much, Diane. That was super interesting. I sure certainly didn't realize that the brain continues to grow throughout our lives. I thought it was fully formed and then we use that for the rest of our lives. But it's good to know that we can do things to affect how our brain grows. I'm wondering, you talked a little bit about exercise and how that can affect the growth of new neurons. What about things like maybe if you get more sleep, or maybe you're in a stressful environment, does that harm the growth of neurons?
Exactly. Most things that we think of as being good for us, so sleeping, doing things like that, those generally all do increase the number of cells. Interestingly, we don't know as much yet about how sleep affects the number of newborn cells. But we do know that animals that are deprived of sleep, for example, for long periods of time, will have less new neurons being formed. Now, stress is another good example. So stress is kind of an interesting thing, right? Because you can have a little bit of stress that could be good for you, but a lot of stress over a long period of time, so chronic stress, like if you have a lot of stress every day in your life, then we know that those kinds of forms of chronic stress do decrease the number of newborn neurons. So clearly the brain is able to respond to what's going on in our life and our environment around us.
Interesting. You talked a little bit also about stroke and the ability to recover from stroke. Does that also apply to diseases like Alzheimer's disease in terms of neurons going in to help with that?
So it actually doesn't. So this is where stroke is pretty unique, actually. So stroke is one of the areas that I research because I find it fascinating that the brain actually after a stroke tries to repair itself. And it does after certain types of injuries. So a traumatic brain injury where you maybe have a big hit on your head, also sometimes new cells will migrate to that region. But in things like Alzheimer's and Parkinson's disease, you don't get that same kind of response. So in Alzheimer's disease, you have a big area of your brain that you don't have as many neurons, but there's no new cells going there. So what we're trying to understand is, can we drive those cells to go there? And why do they naturally not want to go there? So it's like with a stroke and with your hit on your head, it's like your brain recognizes something really bad just happened. And I kind of would imagine like the brain's trying to fix itself. But in Alzheimer's disease, you might know that happens very slowly, right? It's sort of like all of a sudden you start to lose cells very slowly. And it doesn't all of a sudden just have a lot of new cells that are generated.
Okay. We've got a couple of questions coming in from Twitter here. One from, looks like West Point Gray Academy. And they ask, is there a link between diet and neuron production?
Yeah, so this is another, the interesting thing is we've really only known sort of almost about a decade or two that these cells are there. So this is another area that's still really unexplored. And we don't know a lot about how diet affects actually the birth of newborn neurons. So that's something, if you're interested in research, would be a great question to look more into, because that's something we actually don't know a lot on. We do know that if you starve an animal, that would change. So if you didn't allow an animal to eat at all, that would change the number of cells because the animal would respond to that and it would decrease. But we don't know how different types of food would modify it or anything like that.
In the absence of a stroke or a concussion or Alzheimer's, the neurons are still being produced, and they're being produced in the hippocampus, like you said, right?
Yes.
So if there's no other injuries, what are these neurons, what use are they going towards? Just to make us smarter, or?
Yeah, great question. So those cells in the hippocampus are involved in, we think in normal situations like you and I right now, we don't know exactly because we can't look in the human brain when you're learning or I'm learning a new thing, but at least in an animal, we know that they're involved in trying to learn very discrete things. So we know that they're important in terms of learning and memory, remembering new items. So they're part of our learning and memory process. And I didn't have time to mention it today or go into it, but it's quite interesting that the hippocampus is not the only region actually in the brain. So in that brain section I showed you, that was the only region that newborn neurons were being born. But you can also get newborn neurons actually also that go to your olfactory bulb. So your olfactory bulb is part of actually your ability to smell. And we think that those newborn cells in that region, obviously in an animal, that would be really important, right? In terms of their ability to smell. An animal's nose is really big compared to ours when you look at the relative proportion. And in humans, it might be important for us to remember the certain smells of foods or things like that. So it's in learning and memory, both within learning and memory, sort of knew where you parked your car or how you ride a bike, right? But it also might be important in terms of learning new smells and learning and memory in an animal, especially in terms of olfaction.
I see. So if we did have a neuron that went to our olfactory regions, then how would it know where it's going and what to do once it gets there? Right?
Oh, it's a really fascinating event, and that's what we're trying to understand is what are the factors? So what's the thing inside the brain that makes it know where to go? And it's kind of the same kind of questions we're still trying to understand in the embryo. How does one cell know where to go in the end to make the important connections? So those ones that actually go to our olfactory bulb, it's really amazing because they migrate a long way in our brain to find their final destination. So in the hippocampus, they're born right there and they become neurons right there. But the ones that go to the olfactory bulb go a long way. And what they think happens is basically the cells sort of chain migrate. So they sort of crawl along each other in these chains, and then they go right to where they're supposed to go. And we now know that there's a lot of chemicals that surround those chains that try to help them go in the right direction, that basically are chemoattractants that pull them in the right direction to make sure they're going to the right spot.
I guess on more of a process level, if you do have new neurons that are being generated, what differentiates them? Like, how can you say this is a new neuron and this isn't a new neuron?
Right. So I didn't have time to.
Yeah.
Yeah, exactly. I didn't have time to tell you how these fancy mice work and how do we actually see how these cells are. So what makes that cell green, right? So what these mice have is that we know that in those cells there's a certain promoter, so a certain protein that's expressed in that cell, right? So we know that in these cells they have certain proteins inside of them that are only in those cells. And basically what we can do is go into the mouse embryo and the genetics of the mouse and make it so every time that protein is made, it goes green. It allows for the fluorescence signal to come through to be able to see then those cells particularly. So what we do is make what's called transgenic mice, where we can modify those proteins so that they are green, so then you can see those mice over time. But that doesn't really explain in humans how can we tell, right? Because in humans, there's no way we would go in and make that protein green to be able to see. So what actually happens is there's a certain chemical called BRDU. Okay, so BRDU, what it does is every time a cell divides, it gets taken into the cell. So in humans, what's happened is a long time ago, people took BRDU or patients were given BRDU to look at the growth of cancer tumors. And when they did that post-mortem, when they looked in the brain, they could see those BRDU cells within the brain. So they know that those cells were dividing within the adult brain when the patient was given the BRDU.
I see. So that's the way you can kind of transcribe what you're doing with the rats to the human case then.
Exactly. And those proteins that I said that we can make green in the animal, we can also stain for those in sections to be able to understand are those proteins in the cells actually to be able to look at the newborn neurons.
Right. Okay. Probably have time maybe just for one more question. Again on Twitter here. So from Brent Peltala here. How did you get into neurology? What did you do in your educational path to get where you are today? I guess you talked about that a little bit.
Right, so I started out, working in psychology actually, which is a study also of the brain. But then when I went to Nova Scotia, I worked there first in psychiatry for a bit, helping with clinical trials there. And when I was there in psychiatry, I was just amazed by the brain. I couldn't believe that how certain people had, such illnesses that caused their brain to not work correctly, and how very little we really understood about why it was their brain wasn't working right. So that's what got me first pulled into the field of neuroscience or the study of the brain, because I thought this is an organ in our body that's so important, and yet we still know so very little about it when you think of how complex it is. And then when that study in humans came out, remember the study I told you in the late 1990s that said those newborn neurons were in humans and there was a lot of talk? When I read that paper as a graduate student, I was so excited because I knew that this was probably going to make a big difference in what we could do to help people with a lot of disorders. So that then drove me to go and to continue studying the birth of newborn neurons in the brain, the study of adult neurogenesis, to try to really understand how can we change that process so we can try to help more patients and to make lives better in diseases such as stroke.
Well, I can certainly see your enthusiasm for your field, and it's certainly interesting how you can get one major result that changes the course of all research to come, right? And I think this is going to be a very important area of research given the importance that we put onto diseases like strokes and Alzheimer's as we try and learn more about these things. Unfortunately, though, it looks like that's all the time that we have for today. So I'd like to thank you, Dr. Lagace, for your time, and thank you to everyone, again, tuning into our live stream. Thank you for all the questions that came over Twitter, and I'd like to remind our viewers that the next PIR live event will be Tuesday, November 24th, a week from today at 11 a.m. Eastern, when we'll talk with Dr. Mike Eels from Trent University to discuss his work in forensic science and blood splatter analysis. So thank you, Dr. Lagace, again, and thank you to all of our viewers.
Thanks so much for having me.